Hello, everybody. Welcome to today's discussion video. I'm going to be talking to you about cancer, especially in relation to the concepts that we covered in discussion 8, which was the cell signaling discussion, where we saw a lot of those like signal transduction pathways and different ways of adapting to certain like environmental cues or conditions. But now we're going to be like specifically talking about in relation to cancer and like whether cancer is going to develop or not. I would highly recommend watching the discussion 8 video if you haven't had a chance to already. And additionally, you should have noticed that I posted a review video of the like actual answers to the exercise. I highly recommend going over that. Even if the exercise didn't like cause you a lot of grief, I would still recommend going over that because that's going to be a good example of like the types of questions that we'll be expecting you to be able to answer for the sake of the exam. So like I hinted at, we're going to be applying the discussion 8 stuff to an example that hopefully you can see is like relevant to human health. Cancer is like a huge public health issue, causes a lot of like excess morbidity and mortality globally. And so we're ultimately like really concerned about like, okay, what are the different causes of cancer? Why is it able to like affect so many different types of organs? And like, what are the things that are happening that allows this to occur? And so the reason why, like when we're talking about a cure for cancer, the reason why that seems like so elusive or like so out of reach is because cancer is different than other types of diseases. So if you think of some types of diseases like sickle cell or Huntington's disease or something like that, where we have a really specific like gene that's being acted on and something's happening that is like causing normal human functioning to like be depleted in some way, which is why it's going to like cause a disease, like a health effect. Whereas with cancer, it's more of like a general like umbrella term for like a whole bunch of different possibilities. So the reason why this is a little bit harder to like treat specifically is because there's going to be a lot of different ways that we can evade the natural adaptive responses. It's also unique in that it can affect lots of different types of tissues and organs in our body. And even if we have like similar types of mutations occurring at like one site or the other, the ultimate effect on like what that's going to do to our health is going to be like really different depending on what type of organ functioning you're inhibiting, that type of stuff. And another reason why it's a little bit tricky is that like when we have a like singular mutation, like we can think of mutations, they're always going to be generating like at a like single cell level because it's going to occur in this like cell cycle step where we're getting this like the division into like daughter cells and stuff and like replicating. So that's where the mutations are going to initially like start at. And if we think of like an isolated mutation, even if it does cause like some sort of effect that allows the natural checkpoints to be evaded, if it's just like one instance of that, that's not really going to matter to us that much because even though like we have that mutation, it's not going to be inhibiting the function of like some other like important organ in any way. We only really care about this like overproliferation if it's like harming some other part of our body. What is going to be like the dividing line between when we're like just having mutations that evade these checks and balances versus like an actual cancerous phenotype is what net effect we're having like on our human functioning. So we can think of like benign tumors, for example. Those we're going to see this like this phenotype of having proliferated cell growth. But if it's not actually harming us in any way, then it doesn't really like we're not going to say that that's like cancerous because it's not actually like being it's not like having a deleterious effect on our health. It is still something to be concerned about because it's probably going to be more likely that some mutation of that benign tumor is going to be able to like cause it to become malignant rather than if we didn't have that benign growth. But ultimately, like the definition of cancer is just like some sort of effect on our normal human functioning caused by this evasion of these checks and balances. It's going to be really unlikely that there's going to be like a single like catch-all cure that's going to like work at every one of these mutation effects and like at every single like, tissue and organ site. That's just like not really going to work. And so we can think about like why this is so much harder to treat than like a disease that we know that like affects a single thing, and that's just not going to be very likely. So that's why this is going to be a little bit different than just like treating a disease that has like a very specific effect on a single protein, for example. Okay, so overall, I wanted to talk about two different major like regulatory genes that's going to affect whether or not the cancer phenotype is being expressed. Talking about cancer regulation genes, there's going to be two main categories that I'm going to talk about today. And hopefully you remember from like previous units material where we talked about different directions of regulation, how we can have either like activation of something or inhibition of something that causes some sort of effect. We're going to have these two categories that act in opposite directions. And so hopefully you guys should be able to tell me which direction the regulation is acting in, given the following information. So the first thing I want to talk about is the tumor suppressor genes. And hopefully just using the name alone, that should tell you what it does. So tumor suppressor genes under normal conditions is going to do something that is actively preventing this tumor from occurring. So it's probably going to have some sort of function in like either like repairing DNA damage before it continues along the cell cycle. Maybe it'll induce apoptosis. It's doing something that is like acting as a gatekeeper for like allowing the damaged cell to continue and like causes upregulated cell growth. And so if we introduce a mutation that inhibits these types of genes, essentially we're saying if we're inhibiting a suppressor, that's going to be functionally the same as saying that we're promoting cancer. All right, so the second main category is proto-oncogenes. So this is going to be working in the other direction. And so here, under normal conditions, these are actually useful for like a healthy cell. These generally are going to refer to genes that have some sort of role in a signal transduction pathway where like the thing that's being controlled is normal cell growth. So obviously we do want growth to occur, you know, to form structures or to like replicate, whatever. We want cell growth to be able to occur. However, like the fine line here is that if we overexpress these proto-oncogenes, that growth to happen regardless of what signal is being received from like the environment. So this is going to promote a cancerous state because it's going to say we are always going to have these genes being transcribed under this hyperactivation of these proto-oncogenes. And that's going to be the state where we're going to get this like big tumor growth because we're not regulating when growth should stop at this point. And so one thing to note is that like, so proto-oncogenes basically refers to this first step where we're having like a normal function. It's just like playing its role in the regular pathway. When we're referring to this like hyperactivation step where we're promoting a cancer phenotype, we actually refer to these as oncogenes. So it's going to be like the same thing, but now it's going to have a different effect. So that's why we have a different name for each of these two states. And okay, so this was asked on Piazza a little bit earlier, so I wanted to go over it. So to promote cancer, proto-oncogenes must be turned blank on or off, and tumor suppressors must be turned blank on or off. And so we can try to think about, like, I think tumor suppressors would probably be easiest to think about. So tumor suppressors, in order to promote cancer, you're going to have to inhibit the suppressor, and that will like negate the protective effect of that suppressor, which will ultimately promote cancer. And then we remember that proto-oncogenes work in the opposite direction, but even without knowing this part, proto-oncogenes, we remember we said like if they're hyperactivated, that's going to be the part that is going to upregulate this like tumor growth and the cell proliferation step. So sure enough, we can see that proto-oncogenes must be turned on in order to promote cancer, and tumor suppressors must be turned off. Actually, we talked about different checkpoints that are occurring during a cell cycle in order to kind of like check for damage before it proceeds to like the next important regulatory step. And these checkpoints are going to be really important in catching this like DNA damage or like some sort of effect that will cause cancer. These checkpoints allow us to like hopefully catch that before it actually goes into this division stage. So the first checkpoint that's going to be reached is this G1 checkpoint. This is actually going to be the biggest like filter. So most of the cells that are going to be like blocked are going to be blocked at this step here. If the cells are blocked at this step here, they're essentially just going to be returned to their like G0 quiescent stage. And sometimes this will just like wait for conditions to be optimized that will allow this checkpoint to be cleared, or they'll just stay in that quiescent phase. So this G1 checkpoint is checking for just like proper environmental conditions that will allow like optimized cell growth. So like we're looking for an appropriate cell size. We're looking for availability if we don't want to like waste resources growing if we can't like conduct basic functions later on. These different growth factors, we need to make sure that those are available. And then we can also check for DNA damage at this step. If any of those things are not ideal for growth, that's when we're going to see this step being blocked, and we're not going to be able to proceed down the cycle. Assuming that we did clear this first checkpoint, now we're entering this S phase, which hopefully you remember is the replication phase, where we're getting that sister chromatid of the chromosome being formed. And then we're going to hit this next G2 checkpoint, and hopefully you can use like your knowledge of like what's happening at this S phase to kind of like remember what the G2 checkpoint is doing. But so here we had this whole like replication step where we're adding this new chromosome, where we're adding like this new copy of the chromosome. So there's gonna be like a lot of opportunity for potential like damage to have occurred, or like some sort of error, or like maybe replication didn't occur all the way. Like stuff could go wrong in this stage. So at this next checkpoint, we're essentially just making sure, do we have any like signs of damage that we don't want to pass? We already tried to recruit the helper repair enzymes, and there's nothing it can do to clear that checkpoint. Now we're going to signal apoptosis. So even though we made it all the way this far, we still would rather like just say that we wasted those resources and are going to destroy the cell rather than risk moving on. Assuming that we did clear this second checkpoint, we have a third major checkpoint to clear before we can like be finished with this whole cycle. And this is going to be in the mitotic phase itself. It's going to be the spindle assembly checkpoint, which is essentially just going to be checking that the chromosomes are attached to the spindle, and we're going to like have this correct alignment so that like when the daughter cells are formed, we're having like the correct amount in each daughter cell. So we wouldn't want to have like one daughter cell having everything and then one having nothing, because that is going to affect the function. So that's going to be the third main checkpoint here. And so that was just like an overview of what happens. But now I'm going to talk about how these like regulatory like cancer genes can affect this path. So RB is a tumor suppressor gene that works at this G1 checkpoint. And then P53 is another tumor suppressor gene that is really important that works at both the G1 and G2 checkpoints. So I'm going to explain this RB first because it's just working at G1. So if we have a situation where we're having these social signals being present, so social signals is just like another way of saying growth factors. So if we have like some sort of like environmental condition that's saying, hey, we want this transcription to occur in this case, like we want the growth to be able to happen, what's going to happen is the growth factors are going to be able to upregulate the next step in the path, which is this cyclin. Cyclin is going to bind to CDK And hopefully, like, even if you don't know what this stands for, you should be able to say, okay, K is probably going to stand for kinase, because we're in this like signal transduction pathway, and so that's going to be like a major, like, kinases have a lot of activity in these paths. And then we know that in order for this to work, we need to bind to cyclin. So CDK stands for cyclin dependent kinase. And given what we talked about in discussion 8, you should be able to, like, without even looking at the picture, tell me what this bound complex is going to do, because of the definition of kinase. So hopefully you remember that kinases work by catalyzing this hydrolytic reaction that's breaking down ATP into ADP plus this extra phosphate group. And that extra phosphate group is going to be added to, like, the next step in the path, which is going to cause some sort of conformational change that is going to push the reaction forward. So sure enough, when we have this activated CDK cyclin binding, we're able to donate that phosphate group from the hydrolytic reaction to this RB. This is where it's binding, and it's going to cause that this part to be unbound from this E2F subunit. So originally they're bound, and then once this is activated, it's going to release that part. And so even though the binding is occurring here, the thing that we actually care about in terms of, like, pushing the reaction forward is actually this free unit here. So this E2F is actually going to be the thing that binds to these S phase genes and upregulate transcription. So essentially, like, the S phase is not going to happen unless we have this E2F that's, like, free, able to bind, and then initiating the next step of the cell cycle. So, like, in this regular CDK form, the active site is not going to be able to, like, do anything. When cyclin binds to it, we're changing the conformation of the active site. And now we're able to allow that hydrolytic reaction, which is going to cause that the next step to be phosphorylated, which is going to be the activation step for the rest of the pathway. So that's just kind of like a refresher about how structure equals function and why that's dependent on that cyclin. Okay, and so now knowing what that path looks like, we should be able to say which of the following mutations in RB is going to promote cancer. Mutation that prevents RB from binding E2F, mutation that prevents RB phosphorylation, a duplication of the RB gene, and any mutation. So essentially, since we're saying what is going to promote cancer, we're going to see an overregulation of those S phase genes, because that's going to say, like, now no matter what is happening, we're going to, like, be transcribing and continuing on the cell cycle regardless of if that initial signal is being present or not. So, like, we can look at this original picture, and the whole pathway was being activated by the presence or absence of this growth factor. So what if we had a mutation that prevents this RB from binding the E2F? What that's essentially saying is that now we are, like, starting at this step here of the pathway, because we're introducing a mutation that's going to prevent these two things from binding to begin with. So another way of thinking about that is that now we're independent of the things that are upstream of this step. And so this is a problem because now we're not going to be regulated by the presence or absence of this growth factor. We're going to always have this being able to be transcribed, which is going to be upregulated even in situations where that growth factor is absent. So regardless of what's happening up here, since this is always going to be on, we're always going to get this activation, which means that even if these growth factors are not activating this cyclin CDK binding, it doesn't matter because we've already free E2F that's able to bind. So I kind of vaguely said we have these growth factors, which is going to upregulate this cyclin molecule. I didn't really explain why, but we can look here because we talked about it in discussion 8. So in this situation, we're looking at this RAS pathway. Hopefully this should ring a bell. So RAS is like a really versatile G protein that actually is used in a lot of signal transduction pathways. So remember that RAS is like the G protein, the peripheral protein that when it's bound to GDP is going to be inactive, not going to be doing anything. But when this GDP is switched out for a GTP, that is going to cause the RAS protein to be activated. And the reason why it's, like, highly present in a lot of different types of pathways is that having this activated RAS protein is able to essentially, like, activate a lot of different types of effector proteins. Like, a lot of different pathways are going to be able to be activated by this same active RAS protein. This here, these receptors, these are receptor kinases, which you should be able to tell because we have these two subunits that when we have the signal binding, we cause this dimerization to occur, which is going to cause, like, remember that a kinase is a enzyme. It's going to cause this, like, catalytic reaction. So when this binding is taking place, we have this activated kinase that's going to be directly phosphorylating the original GTP, and that's going to be the activation step, this direct phosphorylation. If we had a receptor that wasn't a kinase, it was just, like, doing some sort of, like, conformational change. Since we didn't have that enzyme activity, what instead would be happening is it would interact with this G protein to somehow cause this GDP to be, like, dislocated and then a GTP to be brought in. So I just wanted to bring that up because this RAS protein can be activated either way. It doesn't really matter how we get from GDP to GTP. As long as we get this GTP, we're going to have this, the rest of the reaction being able to be activated, the rest of the cascade. Okay, the next important thing I want to talk about is P53. So this is also a tumor suppressor, but it works at both the G1 and the G2 checkpoints. And its function is to halt the cell cycle process when it needs to be halted, and then repair enzymes when it senses that there's, like, something that needs to be fixed before it can move on. So the reason why the halting step needs to occur is that if we don't have this, then we're going to like recruit the repair enzymes, but then the mutated DNA is already going to be on the next step. So we need to like block it there first, and then recruit the repair enzymes. And then as like a last ditch resort, if after the repair enzymes are recruited and they realize that there's nothing that they can do to like fix this and clear the checkpoint, that is when we're going to say, well, too bad, we're going to sacrifice that cell rather than risk the chance of having a mutation being like passed down through the future daughter generations. Okay, so this is basically acting as a tumor suppressor because it's preventing, it's like natural state is to prevent this cancer phenotype. So we have this like, so under this normal state when there's no damage being detected in the DNA, this p53 is just like naturally going to get out of the nucleon. But when DNA damage is detected, that is going to activate a kinase, which is going to able to, that's going to activate a kinase, which you remember what that does. It donates that extra phosphate group from a hydrolytic reaction and cause that to be activated. These activated p53 proteins are going to be able to bind to the DNA and prevent the next step of the cell cycle to occur, to allow the cell cycle to be halted so that we'll have more time to like try to repair that DNA or signal that we're going to have to induce apoptosis if there's nothing that we can do at that point. So if we have a mutation in the p53 protein that prevents the p53 from recognizing a damaged DNA and preventing that like activation of the p53, that's going to be a cancer-causing mutation because we're essentially inhibiting that protective effect. So we remember that tumor suppressors act in its regular state as a way of like preventing cancer. So when we mutate that and cause it to lose its function, now we are, it's the same thing as saying that we're like inducing a cancerous state, or like at least making you more at risk for a state of cancer. So this slide is asking about, all right, so moving on to the slide here, this slide is essentially just asking us about in a diploid organism, when we have two alleles, so like two copies of a specific like tumor suppressor gene or proto-oncogene, what's going to happen if we only mutate one of them for either the tumor suppressor or the proto-oncogene? Like what effect is that going to have? And this is relevant because it's essentially just like another way of differentiating between how the tumor suppressor works versus how the proto-oncogene works. They're both regulating cancer in some way, but the way that they're acting is different. So we can think of, okay, we have a diploid organism, and let's say that we have we have those two alleles for a tumor suppressor gene. If one of those alleles are mutated so that it's not going to be able to work, what is that going to like do to the overall protective effect of the tumor suppressor? So what would happen in that case is, since we have this one allele inhibited, we're going to lose that protective effect from that allele. But we have that second allele that is still inducing that protective effect because it's non-mutated. So it's able to induce that protective effect, which essentially is saying that we have a fail-safe. So either way, we're going to get that tumor suppressor protective effect happening because we have two copies of that protective gene, but we're still going to be able to protect against cancer. However, this will cause a predisposition to cancer in future generations. So if we're passing on like a daughter cell that's going to have one allele that's already mutated and not able to work, now we don't really have that fail-safe. So now if we have just one single mutation occurring that's going to affect that active allele, now you're immediately losing that function, that protective function. Whereas before, if we had these two alleles, you had like this background like fail-safe in order to occur. So like in order to completely lose function, or like in order to completely lose the effect of that tumor suppressor gene, you'd have to have both of those alleles being mutated in a way that inactivates them both. And so if we're starting out with a case where both of the alleles are functioning, the only way that we would totally lose the protective effect of the tumor suppressor genes is if we just happen to have two of the same mutation arise spontaneously, one in each of the like different alleles. So obviously that is going to be like less likely to happen than if we have this case where one of them is already mutated and then you're just getting one of those mutations occurring in one spot. So this is essentially called a loss of function gene, because remember, when we're inhibiting something from suppressing cancer, that's the same thing as saying that we're inducing cancer. So like a loss of function is going to be like an inhibitor. So now asking the same question about proto-oncogenes. So proto-oncogenes work in like a different direction, than tumor suppressors. And hopefully you can remember that proto-oncogenes themselves are going to be like neutral. They're going to be just like playing a regular role in the signal transduction cascade. But if they're hyper-regulated, then they're going to cause the thing that is promoting that cancer phenotype. So here, if we say that, so like we know that we're diploid, so we have two alleles. If we have a mutation in one of those alleles, this is going to be a gain of function gene. It's going to upregulate this proto-oncogene into just like a mutagenic oncogene. And so even just like having one of those alleles being activated is going to be problematic because that is activating this cancerous phenotype. So here you only need to have one allele being affected in order for that damaging effect to occur. So another way of thinking about this is, if you're like talking about a loss of function gene, you're going to have both of those alleles being mutated in order to effectively inhibit that gene. because the second allele is acting as like a fail-safe and allowing that protective effect to occur either way. But once we're working in the opposite direction and a positive effect is going to induce cancer, even just like one allele being mutated is going to ultimately have that effect. So that's what's going to be the major difference between the loss of function versus the gain of function genes. Alrighty, so doing a quick little knowledge check here. We're told that under normal conditions, BRCA1 and BRCA2 are these like genes that code for a protein that's involved in DNA repair mechanisms. And so using that knowledge, what type of cancer regulatory gene it is, so hopefully you should be able to take a second to predict. And since we know that like in its natural form, it's protecting against a cancer phenotype, that's going to say that this is going to be, that these are acting as tumor suppressor genes. And so the trick here is just to look at like, what is it doing in its natural state? The type of mutations that you can have in proto-oncogenes that will cause that hyperregulation to occur. So looking at this should remind you of discussion 8. So you have these kinase receptors here. We have these two subunits that we see this dimerization occurring once the signal is bound. And that is going to allow the activation of the very next step in the cascade. A mutation that can cause this proto-oncogene to be hyperactivated into an oncogene, the gene state that's harmful and cancer causing, is this mutation here, where now we can see that this dimerized form is always going to be activated. So again, anything that's upstream of like the initiation point, or like the thing that is going to be always activated, is no longer going to be like a regulatory portion of that pathway. So in this mutation, now we're saying that we're no longer regulated by the presence or absence of this initial like signal, because this is always going to be on regardless of whether that signal is there. And then that is going to cause this hyperactivation step of the proto-oncogenes, because when this is always activated, it's going to push to the very next step of the reaction, and then every other reaction after that step, since they're all downstream of each other, that's going to cause this system to always be activated. And activating too much of these growth factors is going to be ultimately damaging and like cancer promoting. Okay, and so now I actually want to just like go over some lecture slide pictures. Let's say that 10% of a certain type of cancer has overexpression of the receptor kinase that activates this RAS-MAPK pathway. What is something that we could do in order to treat this condition? Now, I would try to pause the video and try to predict. This is where like going over that discussion 8 exercise video is going to be really helpful, because this is like the situation where we're trying to predict what situation, like where in the pathway can we target to cause like a certain effect to occur. So we're trying to reverse this overexpression, given the fact that we know that this is the step that's being overexpressed. Okay. So we can look here. So essentially, we were told that this that the thing that's being activated at all times is this receptor kinase protein. So what we can do to effectively inhibit the rest of the reaction is we can add these like receptor kinase antibodies, which are going to bind to the activated receptor kinase sites and essentially just like neutralize them so that they're not able to be constitutively active, which will prevent which will prevent the activation of all these other steps. And we could also have a inhibitor that's going to just like compete with this ATP for the binding sites. That's also going to be another way of preventing the rest of the cascade from occurring. Alrighty, so on this slide here, we're talking about a mutation in the RAS oncogene protein. I want you guys to focus on this second bullet point mostly. So we're being told that if we have a missense mutation at amino acid sites 12 or 13, what that's going to do functionally is it's going to like cause this GTP being bound, and it's never going to be able to have this extra phosphate being cleaved back into its GDP form. So hopefully this should remind you of something. So in discussion 8, we talked about this GTP gamma S analog that essentially had like the same like net effect. It was never able to return to its GDP state. It just like worked differently. It wasn't because of a missense mutation, but either way, we were getting that same net effect. And we can remember that the whole like gist of the RAS oncogene or the RAS gene is that if you are bound to the GTP form, you're always going to be on. If you're bound to the GDP form, that's when you're going to be off. So this is going to, this mutation is going to be like an oncogene hyperactivated state, because now regardless of what signal is being received, you're never going to have that gene being inactivated. So you're always going to be pushing the cascade forward, regardless of whether or not the cell signal is actually like telling you to do that. Okay, so we're given this pathway here, and we're told 15% of non-small cell lung cancers have hyperactive RAS-G proteins. So essentially what this is saying is that we're now changing the initiation step to this point here. Before the initial, the other pathway that we were just looking at earlier was saying that this was the step that we were hyperactivating. Now we're hyperactivating this step. So it's always going to be like the things that are downstream that are going to be able to occur. So now anything that's upstream of the constitutively activated part is not going to have any effect on whether or not the reaction can proceed. So when we're asked how could we treat this, essentially we're able to eliminate this step here as being a possible like mechanism of action. Because even if we added an inhibitor here, it doesn't really matter because we are going to have this always activated and then the cascade could occur. So we're essentially just like using the process of elimination to prevent or like to know that this is not going to be the target in this pathway. Okay, so now moving on, this is going to be a little bit of a different mechanism than we've seen before. And I don't want you guys to like worry too much about what like each of these chromosomes are and like what each of these sites are and blah, blah, blah. I want you to focus more on like the mechanism of how this is serving to like promote a cancer phenotype. So now on this slide, we're told that this cancer is going to be caused by a translocation, so like a flipping of the position between these two different sites here, the IGH and the cyclin. And when that translocation happens, now this cyclin is going to be really, really closely bound or like closely associated to the enhancer sequence of that IGH. And since that is going to be like really tightly associated with that now, and the enhancer is always going to be upregulated, we're also going to have this upregulation of the cyclins. And so trying to remember from my discussion slides, we said that when we have those cyclins that are upregulated, what that does is it's going to allow like the rest of that signal transduction pathway to another mechanism is via methylation. So hopefully you can remember that when we have this methylation occurring, what that's going to do is it's going to cause the chromatin to like condense really, really tightly to each other. And what that means functionally is that there's not going to be like room for like the transcription factors and like the regulatory proteins, all that stuff. So we're physically blocking any sort of like gene transcription at that point from hypermethylation. So if we're hypermethylating and blocking the transcription or the expression of a tumor suppressor gene, trying to predict what that will do, that is essentially going to be the same thing as saying that we're promoting cancer because we're silencing that that protective effect, that tumor suppressor effect. And we can also think about like the opposite reaction. So hypomethylation is going to cause like a really like relaxed structure from that that initial supercoiled form. We're going to have a lot of extra room for like all this like transcription and like gene activity to occur. And so this is going to like upregulate any genes that are located on that site. And so what that's going to do is it's going to like from its natural state, it's going to be upregulating transcription. And so if we have proto-oncogenes that would function normally, but now we're increasing transcription, that is essentially saying that we're transitioning from that proto-oncogene like neutral state into something that's going to promote a cancer phenotype. Another type of mechanism that can happen that would promote a cancer phenotype is with telomeres and telomerase. So hopefully you remember that like under normal conditions, act as kind of like a buffer like protection sequence against this phenomenon that we see like every time we have this replication fork here, the lagging strand is always going to be shorter than this leading strand because of this limitation that we have to be synthesizing 5' to 3'. And so we have those original like primers that were added there to make those Okazaki fragments that have to be removed. So the DNA polymerase one is going to come and remove that, but we're not going to be able to synthesize. We're not going to be able to like restore that additional length because you still have to conform to the 5' to 3' direction. So if we didn't have telomeres, what would happen is like at each step, this original like leading strand would stay the same, but each step would cause this lagging strand to become shorter and shorter and shorter. Which obviously would not be good for the sake of like creating new strands based on a template. So when we have just like a normal telomere, since these are non-coding sequences, we're still gonna have that phenomenon where each step is going to have this like subsequent shortening of the lagging strand, but it doesn't really matter because we're protected by these like non-coding portions. But we're gonna eventually reach a point where we don't have any more telomere to protect against DNA damage. And so at this stage, we're essentially gonna be reaching a signal that's telling these cells to stop. It's going to say, you know what, we're like reaching the end of our lifetime. So this is going to be like the end of our replication cycles. When we have a cancer cell, there's going to be this telomerase enzyme that is essentially just like going to be immediately reversing the effect of. And why that's relevant is that since we're immediately just like restoring the original length of the telomere, we're never going to get to a stage where we're reaching the signal that's saying, hey, we need to stop replicating at this point. It's like when we're thinking in terms of cancer, we don't want to have immortal cells because that just means that you're going to keep growing and growing and growing and growing. And when you're talking about a tumor, that's not exactly something that you want. All right, and one of the last important factors for promoting cancer phenotypes is this angiogenesis, which I was talking about earlier, this creation of these like extra blood vessels. So here's like a normal condition where we see the blood vessel is like acting at this side here, and like it's essentially just like causing oxygen and like important like resources to be spread. Like when we have this tumor growth, eventually we're going to get so big that the actual blood vessel is not going to be able to like reach this top end of the tumor. And so one unique pathway of these cancer cells is that they're going to be able to detect when this environment is like low in oxygen, and it's going to stimulate the production of this like HIF transcription factor. This transcription factor is going to serve to recruit another enzyme called VEGF. I don't think we even know what that stands for, but yeah, that transcription factor is going to cause this VEGF to increase, and that is going to be the thing that's promoting this like new blood cell creation. It's like using what we know about how this angiogenesis is occurring. We're told down here that anything that's going to inhibit this VEGF is going to be used to treat the forms of cancer that are like really relying on this mechanism in order to grow. And we can see that here. So like if we inhibit this VEGF from occurring, even when we have the high transcription factor. If we're never going to be able to express this, then we're never going to be able to activate this next step, which is going to be the angiogenesis. If we had another inhibitor that would protect against this step here, so that like low oxygen wouldn't affect this transcription factor, that would be another way that we could potentially treat, like prevent this angiogenesis. But I'm assuming that something about that is not super functional because it doesn't say that we have like drugs that do that. So anyway, anything that's going to be like blocking this pathway from occurring all the way down to the net effect is going to be a potential target for treating cancerous cells that rely on like this specific route. Okay, that's all I have for you guys. So with the exercise and the quiz, I feel like a lot of the concepts I like talked about directly during this video, so it should be a fast exercise. If you guys have any questions still, let me know. I'm going to have my office hours as usual, Fridays 3 to 4. Exam is coming up just next week. I know it's coming up super fast, so that's why I recommend taking some time this weekend to work on like the problem roulette question, kind of like listing out the things that you're still having troubles with. I do believe that the course evals for the class are up. So if you go onto Canvas, the section site, you should see a tab on the sidebar called Teaching Evaluations that you're able to click, and it should automatically take you to the survey. Please make sure to fill out this for me. So like make sure that you're not just doing the lecture, the teaching evaluation on the lecture site, because that's going to be all like given to Lori. And I really want your feedback too about how I can possibly improve for future semesters. So I'd really appreciate if you guys could take a minute or two to fill those out, especially because this is our first time doing a fully virtual class and kind of going an unconventional route of having a asynchronous and synchronous option. I'm curious to hear your guys' thoughts on how that worked for you, and if you would suggest something different for the future, so we don't really know how long this virtual only system is going to be in effect for. So I really would like to hear you guys' opinions and feedback. And I believe that Lori is offering 3 extra credit points for people that do both the lecture and the section surveys. So please take a minute to do that for the sake of those bonus points and also for me to improve. And alrighty, if there's anything in the meantime that you guys want to talk about, feel free to email me, or we can set up an appointment to do like a Zoom call or something if you want to do some more in-depth like face-to-face review with me before the exam. Yeah, keep in touch if there's anything I can help you with, and otherwise I will see you next week for our final discussion section of the semester.